So a couple of announcements before we get started. The first one is hopefully all of you got an email or a link to a review for the class, what you thought about me, what you thought about the class. There's nothing specifically in there about clickers, but please let me know. Great, horrible, waste of time, absolutely wonderful, force me to reread the lecture before class. Yeah, so please give me some feedback on that, because that's one of the things, and we're just discussing engaged learning up here at the front. What you think about that, what you think about online quizzes, etc. So there's a free text way you can put in your opinions about that, and it's all anonymous, so I don't know who it's from. Of course, the test is all anonymous too, because you just have your student number, and that's happening when? When? 8 a.m. exactly, right here. So that's the exam. I mentioned also that there was a problem with my mic that happened at the end of the lecture on Wednesday. Batteries died. I guess these things happen. I checked it before lecture. I thought they were okay. So if anybody's got an audio recording for that second half and can send it to me, that would be great. Otherwise, what I may do, and if you're interested in audio for those kinds of slides, I also have all of the lectures from the previous term on there, and a lot of the things there are pretty similar. So at least you can check out some of that. And I'm working on getting an audio for Eric Iverson's lecture, and we're going to talk a little bit about some of his things today in lecture as well. And again, as I've hopefully probably mentioned ad nauseum, there's nothing new, at least in terms of slides in today's lecture, but I noticed going through things that I thought there were a couple things that I did a really crummy job explaining the first time around. So this is good. It gives me a chance to go back and talk about them again this time. So, and I put brand new batteries in here, so that should be working as well. So let's get started. Last two clicker questions of the term. Sigh. To detect a few proteins on a 2D gel, which had a ubiquitin added, you would do a Western, Southern, Northern blot, mass spectrometry analysis, or NMR spectroscopy.
What did you put in your notes? We have 15 seconds.
15. 10. 3, 2, 1. Liftoff. So, yes, it is a Western blot. Yay, hallelujah. What kind of antibody would you use to do your Western blot with? The antibody would be reacting with what? Ubiquitin. So it would be an anti-ubiquitin antibody that you use for your Western blot. Western blots are used to detect proteins. Southern blots are used to detect DNA. Northern blots, RNA. So, and Southwesterns? DNA binding proteins. They actually really are called that, yes. That too. So our next clicker question, we need to select the right answer here so people actually get points. They would probably appreciate that, which reminds me, I will be posting clicker scores as soon as I get done with office hours today, which may be, 8 p.m. or something. But so our second clicker question for today is after 3 rounds of PCR, how many double-stranded DNAs of correct length do you have? 0, 2, 4, 8, or 16?
But that's the correct length.
15, 10. Okay, what did we think? Let's show our results. Split about half and half between 2 and 8. Those of you who have to leave can disappear. I'll be discussing why the right answer is happening right now. The correct answer is B. Yay, those of you who got it correct. And this is all about how many of the correct length you have. And the reason for that I'm actually going to show on the next slide, because this is obviously a confusing thing. And this is also one of those slides that when I was putting this lecture together, I thought to myself, I didn't do a very good job of explaining this. So let's take a look at that. That's here. And no, it's not the video. Okay, take-home message from here. We go over to this side, the 2 ones that are highlighted in yellow. So that's it. That's why that is the correct answer. But the real question is why? Why is that the correct answer? And that has to do with the fact that what you start with, this DNA right here, is much longer than the piece you are trying to amplify. So you separate these 2 strands, and what I forgot to mention, I think, last time, is how do you separate those strands? You just heat them up. So basically, to have this, you need what's called a thermal cycler. Just raises and lowers the temperature all the time. And so when you raise the temperature, the 2 strands come apart. You lower the temperature. Then you get your primers which bind. And these are primers which you have usually ordered from some company which literally synthesizes them. So they make these primers for you. And you have a whole bunch of these primers around. So with these primers, they will now bind to one of the 2 strands. And then you put in a DNA polymerase. And really important for this process, if you don't want to add DNA polymerase each time, is you have a thermostable DNA polymerase, because you're heating up this reaction every single time when you go through another cycle. And so the discovery of DNA polymerases that were stable to these high temperatures was really critical in terms of being able to allow PCR to actually be useful. And where did they find them? Actually, not viruses for a change. Volcanic hot springs in Yellowstone National Park. And do you know how much money Yellowstone got for that? Absolutely nothing. And it's a hundreds of millions of dollar business right now. So yeah, there's some issues with that we can, again, talk about after the final. But yeah, so you have a thermostable polymerase, and what it does is it will extend from these primers. It extends from these primers and will just keep going until it falls off. And so these products that you make here are much longer than the piece that you want, which is right between those 2 primers. So after the first round of PCR, you just have what you started with and these other products. Again, you heat them up, they come apart, the primers come down and bind to them, they extend, and now, after the second round, you've got a couple of pieces that are the right length, but their double-stranded with their template, which was much longer. Yeah?
So when you, in the question, I was confused. I was thinking 2 or 3 full rounds of this, but when you say rounds, that's analogous to cycle?
Yeah, so round and cycle.
Okay.
So that may have been part of the confusion. So that was me, people who had the 4 in that case. And if there's a question like this in the exam, I'll try and be absolutely clear about it. So again, the reasoning now, again, we've still got our longer pieces here. It's only after you do that 3rd set of heating up that now you have a template which is the right length. And so when you extend off of that template, you end up with a double-stranded piece that is the right length. And this process will continue to give you pieces that are too long, but the pieces that are the right length are going to amplify exponentially, and the pieces that are shorter only arithmetically. And so you have to do on the order of 20 or 30 rounds of PCR before you get just, or not just, it's the vast majority of the bands that are the correct length. Does that make more sense now? Hopefully it does. Yeah.
So is it after those first 20 or 30 rounds that you start to see the exponential increase?
Well, so the question is it only after those first 20 or 30 rounds you have to start to see the exponential increase? The answer to that actually depends on how much you start with. Right. If you start with 1, it depends now on how sensitive your detection technique is. So if you've got a really sensitive detection technique, it's even after this point, after the 3rd cycle, then they'll be picking up exponentially. Because it goes to 2, to 4, to 8, to 16, et cetera. So, but again, it just depends on how sensitive your technique is for using that. And there's something called quantitative PCR that literally does that and measures that. But that's a different story that we don't have time to talk about today. But take recombinant DNA techniques next term, and I'll talk all about it. Okay, so backing up a little bit, way back to lecture 17, which is where we started this section of the course. We mentioned microarrays really briefly, and this is also something that is covered in the techniques chapter a little bit, chapter 8. Also was covered back at the beginning, I guess this is chapter 7, I think, beginning of chapter 7. But the idea here is really it's all about hybridization. And so the idea of being able to pull 2 strands apart and then put 2 strands back together with the specific piece of DNA that you're looking for. And the way that this is done is actually with cDNAs. So it's good review for cDNA. cDNA is what? Complementary DNA. So it's a DNA copy of the RNA that you started with. And the way that you get the labels here, so you get the red versus the green, is when you do that complementary DNA, making the DNA copy, you include one of these non-radioactive nucleoside analogs in the process. So you can detect it. And these are usually fluorescent. And so as you're doing that extension, you're making the DNA copy, you're including a fluorescent label. And so you get here, you've got 2 different conditions that you look at. One where you make your cDNA library and label it red. The other condition, you make your cDNA library, label it green. You mix the 2 of them together, and then you do hybridization to everything that's on your microarray, which is usually every single gene in a genome, or something that you're interested in. And those will then either be green if that gene is expressed in the one condition. It'll be red if it's expressed in the other condition. If it's expressed in both, it's actually going to be either yellow in the normal case, or here they've actually just shown it as black. So each of these are just representing, and each of these columns actually represents 1 microarray from 1 patient. So it's 1,800 genes in this particular case, and you're looking at those microarrays as they're hybridizing to them. And that tells you that the RNA expression pattern, in this case, from a bunch of different cancer genes, is different. Yeah?
So you're hybridizing it to mRNA?
So the question is, what are you hybridizing it to? So usually in this case, you'll be hybridizing it to a genomic DNA library. So you've taken the whole genome, you split it up in little pieces, and each of those individual pieces is 1 spot, or 1 gene in this case, on your microarray. And it's usually DNA, because DNA is a heck of a lot easier to work with than RNA. You can do RNA arrays, but they're a drag. So this, I just wanted to pop up here as a great place to think about reviewing when you're thinking about the exam, because basically we've talked about all of these different processes. If you're into doing diagrams or what are they called again? Thought maps? Concept maps. This is a great way to start. It would be to have this right in the middle, because that's really everything that we've talked about. Didn't bring my DNA model because it's got all kinds of problems with it, but just wanted to remind you that almost all of our gene regulatory proteins, which are present at very high numbers of genes but very low concentrations of proteins, almost always going to be binding in the major groove of DNA, because that's where you have the methyl groups that you have on thymidines. You'll have all the hydrogen bond donors and acceptors, which then the amino acid side chains can interact with. And so you can get all of the sequence information that you need to be able to bind specifically to DNA, all looking in the major groove. And most of the time, that's going to be an alpha helix, which is sitting down in the major groove because the side chains are sticking out around the sides of that alpha helix. So that, again, is almost all of these gene regulatory proteins. I wanted to revisit this cooperative binding again, because I also didn't think I did a terribly good job of explaining it the first time around. Basically, the idea here is you have kind of an on-off switch, and that's dependent on your protein concentration if you have some kind of cooperative binding. And those of you hopefully remember some of your chemistry. This is basically like a two-step reaction as opposed to a one-step reaction. Because what's happening here is first you have interaction between, in this case, a heterodimeric DNA binding protein, green and blue. That's your first reaction. And then the second reaction is binding to the DNA. And what that means is at a low protein concentration, you're just going to have these monomers because they're not going to find their partner to interact with. As you move to higher concentrations, now they become a dimer and bind to the DNA. And this will give you a much sharper increase in DNA binding than you would if this was just sitting there as a DNA binding protein here, stable homodimer, a heterodimer all the time. Does that make sense? A little more? So at low protein concentrations, you don't have binding. At medium concentrations, it actually becomes completely bound. And so it's really much more about a switch, on or off. And hopefully, when we get to some of this stuff, it'll become obvious why that's important too, to think about bacterial transcriptional regulation and some of the other transcriptional regulations as well. Electrophoretic mobility shift assay. This is probably what we've most discussed in office hours and on various emails and things. Just wanted to go over it reasonably briefly here. What we're looking at is the shift, down here, between this unbound DNA and something which is slowing down its mobility. And in this particular example, we've got lots of exactly the same DNA fragment, and again, we'll just call this a promoter, and then relatively small amounts of these different protein complexes. If you put all of these together, some of the DNAs will be bound to this protein, some will be bound to that protein, etc. Each of those would give you one different shift. Some of these could be complexes of proteins. That would not be surprising at all, that there are multiple proteins that are involved here. And then each of these lanes on your gel is going to be one fraction from some column chromatography. Be ion exchange, gel filtration, even for that matter affinity. It's more likely to be ion exchange or gel filtration. And so in each of these fractions where you're separating the proteins, they will have a DNA binding component. So here we just have the DNA binding component, something that shifts the DNA to this position, and so on and so forth. So this is the way that practically all DNA binding proteins, certainly the early ones, were isolated. Was doing a column chromatography assay. Some people ask me about how do you measure activity, little blue peaks, if you think about some of those slides. Very often it's going to be something like this. So an electrophoretic mobility shift assay. People questions on this? Yeah.
Having multiple of those proteins bound to a single DNA. So that would show up, I assume, as a different band.
Okay, so the question here is if you have multiple proteins bound to the same piece of DNA. So say, again, for argument's sake, C1 here is a band which is due to a complex of proteins. So this could be multiple different proteins that bind together and then bind to DNA. So a lot like we had with our cooperative binding right here. So if you only have binding when you've got these two proteins together, you'll get that shift, and that particular shifted band will contain the DNA and two proteins, again, in this particular case.
I'm thinking of like we have the C1 complex of proteins and a C4 complex of proteins both bound to a single strand of DNA.
Yeah, so bound to one of these DNAs here. Yeah, so that's why I'm mentioning you've got multiple copies of DNAs here, and so you also have a vast excess of DNA over protein here. And so you should be getting those individual ones. And sometimes you'll have some that have more. So one of these bands could actually be due to C1 and C3. And in fact, you do see a band up here at the top, which could be both of those then binding that same piece of DNA. But you always are going to have your DNA in excess, which is down here at the bottom. So that's one of the keys to this. If you had a really small amount of DNA, then all of these proteins would all be bound to one fragment, and you just get one big blob up here at the top. Does that make more sense, hopefully? Yeah? Okay, so the question is, would you use, and I'm going to paraphrase your question a little bit here, sorry. Would you use SDS page for something like this? The answer is no, and why? Why would you not want to use SDS for this? What does SDS do? It screws up the charges, which is one thing, but the other thing is a detergent. It helps you denature the protein. If you've denatured your protein, is it going to bind to DNA anymore? Highly unlikely. Yeah?
Is there a limit to how long of a DNA fragment you want to use in this kind of an experiment? So you're testing for like a single gene or a single region of DNA?
Yeah, so the question is, there's a limit to the length of DNA that you want to use for this, which is, it's a great question. It's not so much, how much and how many binding proteins you have. It really has to do with the gel electrophoresis, because what you're looking at is a difference in mobility. So if your DNA piece is too big, then any kind of difference you're never going to be able to see. So you want to have a relatively small piece of DNA so you can see the difference with the shift that you get with a protein binding to it. So it's usually on the order of a couple of hundred, between 50 and a couple of hundred nucleotides. And that is actually a great review question, because we may or may not get to gels at the end of this review. What kind of gel would you want to use for this? Agarose gel, acrylamide gel? Acrylamide gel are higher resolution, agarose gels are much lower resolution. Probably going to want higher resolutions. These are almost always done in acrylamide gels. So I will now transmogrify myself, grow a couple of inches, lose a lot of gray hair for Eric's lectures. So this is my review. Again, these are some of his slides there. I just wanted to mention here there's sort of really two kinds of regulation that happen in bacteria. Yes, there's more than that, but the vast majority of these two different kinds, which are going to be positive regulation and negative regulation. So positive regulation is an activator, negative regulation is a repressor. So repressors can still activate genes by the absence of a repressor. And so we talked about this a little bit in office hours. It's all double negative. So if you repress a repressor, that ends up giving you expression. So this is a really nice case here of exactly what's going on there. We have a repressor. It's the trip repressor. And that trip repressor is going to bind to the operator and block RNA polymerase binding when there is tryptophan around, because this is a tryptophan biosynthetic operon. And it's actually really big, make lots of proteins. So we know transcription's energetically expensive, translation's even more energetically expensive. So you don't want to be making all of these biosynthetic genes to make tryptophan if you've already got it. It's really kind of a waste. So the way this is regulated is in the presence of tryptophan, your repressor will bind to the operator and the polymerase can't bind. Of course, in the absence of tryptophan, it doesn't bind, the RNA polymerase binds, and you transcribe all these genes because you need tryptophan to be able to survive. So this is a nice case of negative regulation. The Lambda repressor is unfortunately really nastily named because it turns out it's a repressor and an activator. So it acts both negatively and positively. The positive aspect is, in fact, giving you a positive feedback loop. The Lambda repressor helps binding of the RNA polymerase to its own promoter. So it's a nice positive feedback loop. And most of the time, we'll talk about now, we'll talk about later, how are these activators working? They're bringing the polymerase, they're bringing the machinery to the correct promoter. The flip side of this, of course, in Lambda is the CRO, which is a repressor, and it's just a repressor. It doesn't act as an activator, which will block expression of the Lambda repressor genes. Sometimes, and again, I'll try to avoid this in exams, Lambda C1 is just a different name for the Lambda repressor. So Lambda repressor equals Lambda C1. And we'll talk much more about that next term in virology. So the classic example of how you put both negative regulation and positive regulation together is in the lac operon. Now, the lac operon is fundamentally different from the trip operon because in the trip operon, it's a biosynthetic operon. It's making tryptophan. Here, this is a catabolic operon because it's breaking down that particular substrate. So now in the presence of that substrate, in this case lactose, you want to be expressing the genes, as opposed to in the presence of tryptophan, not expressing the tryptophan genes. So here we have a repressor, the lac repressor, the little orange square here, that is going to bind in the presence or the absence of lactose. Yeah. So the repressor is going to bind and block transcription when you don't have lactose. Because what's the point of making these genes if you don't have something for them to work on? So the other thing here is if you've got lots of glucose already in the cell, the product of the beta-galactosidase, which you make from breaking down the lactose, is glucose. If you've got lots of glucose, there's no point in making these genes either. And so that's where you have our combinatorial regulation. We have our activator, which is the CAP protein, which will activate in the absence of glucose, but only in the presence of lactose will you get transcription of this gene because of the lac repressor protein. In the absence of any of these things binding to the DNA, you'll get a little bit of transcription. As Eric was saying, remind them this is a leaky, this is what you see, leaky. It's a small amount. It's not great, but you'll still get some transcription and some expression of those genes at the at the end there. And that leads to probably the second most, oh, sorry.
I just had a question about that third one down, when there's no lactose and no lactose.
This one?
Yeah.
Mm-hmm.
And it seems like there would still be no transcription happening at all, and then how would the cell be able to process any of those sugars? Or how would it survive if it couldn't break down, if there wasn't any lactose around and it couldn't, and there wasn't any glucose either?
Oh, so yeah, if there wasn't glucose and there wasn't lactose, there better be some other sugars around. If there aren't, it's going to have trouble. So yeah, that's, and if you, again, sort of transfer this, say, I don't know, there's, trying to think of another disaccharide, sucrose operon, for instance. E. coli doesn't work with sucrose. But just say in some organism there'd be a sucrose operon. And so that would be interacting, the repressor there will be interacting with sucrose as opposed to lactose. And so hopefully if you didn't have lactose, you'd have sucrose, and then you could grow on that, for instance. So there are these different operons in the different genes. Okay, so that brings us again to the second most asked questions, I think, in office hours and online, and that is this use of the lac operon in molecular biology. There are a couple of aspects here that are really quite important. The first one is that you're using a, of course, viral RNA polymerase, the T7 RNA polymerase. T7 RNA polymerase recognizes completely different promoters than your normal E. coli polymerase does. And so that way you can have your favorite gene, which is in a plasmid, which we talked about last lecture, which has a promoter for this T7 polymerase on it, T7 RNA polymerase. That gene will only be made when you have this T7 RNA polymerase around. That is regulated through the lac operon, which has been stolen. The regulatory sequences are put in front of that T7 polymerase. And so you can induce the production of your T7 polymerase, and then by extension your favorite gene, by putting in lactose, which is one possibility, but it turns out lactose is not a terribly good substrate because it gets broken down because the cell has beta-galactosidase. So what you use is a non-hydrolyzable analog, this isopropyl thiolgalactoside. Yes, say that 58 times quickly. IPTG, which is why everyone uses the acronym. This then binds to the lac repressor, has it come off the DNA, so the lac repressor thinks that there's lactose around. You get expression of the T7 RNA polymerase gene, which leads to really good expression of your favorite gene over here. And so it's how the technologists have taken advantage of knowing something about viral biology and what the promoters are for that, and also using the lac operon and taking advantage of that lac operon in order to get expression. Yeah? How do you get the T7 gene into your favorite organism in the first place? There's a thing called DE3 right here. Turns out it's a defective virus that has that polymerase gene on it under control of the lac operon. So this is an engineered cell which has an engineered virus in it, which you then add the plasmid to. So it's multiple levels of engineering that you do to make this Franken coli, which will now be expressing your favorite gene. And if you grow more than 10 liters of it, you have to have a special approval from the Institutional Biosafety Committee, whose chair is standing in front of you right now. So, if you want to do that, let me know. Yeah.
One more quick question about that last slide.
Yeah.
Why do you, if you're inducing IPTG so that the lac repressor is always dissociated, is not blocking expression,
Mm-hmm.
then why would you also express the lac repressor? Why not just block that protein from being made?
I'm not sure I quite understand the question. So basically, why do you have two lac repressors here, a lac repressor binding site here and a lac repressor binding site over here? Or
Why would you make lac repressor in the first place?
why would you make lac repressor first? Because if you don't have the lac repressor here, you'll be making the T7 RNA polymerase all the time. Because we just mentioned last time, if you don't have that protein, you'll have leaky expression. So there'll be a little bit which is being made. The T7 RNA polymerase is a really, really good polymerase. It'll make a ton of RNA of your favorite gene. If your favorite gene happens to be toxic, or the poor little Franken coli is trying to do anything else like grow, then you could be causing all kinds of problems. Yeah?
So using the lac operon as kind of a repressor for your for the T7 gene, that's kind of a safety so that you can allow your Franken coli to replicate and get a bunch of them before you start expressing your
Yes, exactly. So the idea is that you make a lot of your E. coli and it's happy, and then you flip the switch and it's really unhappy. And the way you flip the switch is just add IPPG. Aren't they smart, these genetic engineers? It only took them 20 or 30 years to develop it. So you've made your favorite gene, and it happens to be a gene regulatory protein, which binds to a regulatory sequence in a promoter for a eukaryotic gene, because who cares about bacteria? So this is that overview of what goes on at a typical, which of course means probably none of them actually exactly look like this, eukaryotic gene regulatory sequence. So I wanted to mention this a couple times. First one is that when I talk about a core promoter, and you'll probably see this again and again, not just me, that's where your RNA polymerase and all of the general transcription factors are going to bind. And it's just labeled here as promoter, but most of the people will also call this the core promoter. That's nowhere near enough DNA sequence to get the appropriate regulation of transcription at any given gene. And that's where you get all of these other gene regulatory sequences. Most of these are going to be bound to enhancers. So again, enhancer is a DNA sequence that you then have proteins that bind to. These can be tens of thousands of nucleotides away, and usually this whole complex, all of the regulatory genes are going to be regulatory sequences, I should say, on the order of about 50,000 base pairs. So it's big. There's a lot of DNA which is there, and a lot of the junk DNA, because it's not coding for protein. It's just where you have binding sites for various different regulatory proteins. The other thing that I wanted to just emphasize here, we've got about 20,000 genes, protein-coding genes in the human genome, but so far about 70,000 promoters. We don't know what about 45,000 of them do, but they're definitely there, and that's where we've got RNA polymerase binding, et cetera. The way that was found out, by the way, was chromatin immunoprecipitation. So if you look where RNA polymerase and the general transcription factors bind, there are about 70,000 of them in the genome. So how do you get this activation? It can happen either directly with interactions with your RNA polymerase, directly with interactions with your general transcription factors, or through some kind of co-regulator. That's the mediator complex, this big purple thing here, but it doesn't have to be via the mediator complex. Co-regulators, again, can interact directly with the polymerase, or very often they're going to be interacting with chromatin. You'll have those kinds of interactions, and we'll look at those in just a second here. But basically, what do you need to do to get transcriptional activation is get the RNA polymerase to the right place. And usually, your chromatin is bound up in such a way that it's hard for the polymerase to get there, and it's those gene activator proteins that are helping bring everything to the right place. Yeah? What is a holoenzyme? Thank you for bringing that up, because that was something I was just about to mention because I forgot to mention it the first time around. So the idea of a holoenzyme, and I'm not sure if Eric talked about this, but in bacteria, the holoenzyme is the RNA polymerase plus the sigma factor. And in eukaryotes, it's going to be the RNA polymerase plus all the general transcription factors. And so it's that complex which is associating with the DNA. And we talked about when you get transcriptional initiation in eukaryotes, you've got TBP that binds with TF2D. Then you have TF2B binding. Then you have TF2E. Then you have TF2F. Then you have the polymerase. Then you have TF2H. Well, a lot of times all those things are kind of together. And so you'll get just the holoenzyme binding. So at different promoters, sometimes you get the whole kit and caboodle binding at once, and sometimes you'll have the individual ones one after the other. Yeah?
So is core promoter and holoenzyme synonymous in eukaryotes?
So is core promoter and holoenzyme synonymous? So the promoter is a DNA sequence. The holoenzyme is the protein complex that'll be binding to the core promoter. Yeah? So here, again, people will argue about whether the mediator is part of the holoenzyme. Here they say that it is. I probably would say that it isn't, which would mean on an exam, it would be like, okay, it's going to be one or the other. I'll try not to have it, again, if it's controversial, I'll try not to have a question on it particularly, because hopefully the knowledge you get here will be something you'll be carrying on, and if it's just something that Stedman thinks, who cares? In the back.
Is there a difference between the holoenzyme and the pre-initiation complex?
So the difference between the holoenzyme and the pre-initiation complex, the pre-initiation complex is promoter plus the holoenzyme. So that would be your pre-initiation complex. Yeah?
I also just want to mention, I spoke briefly with Dr. Bartlett.
Mm-hmm.
He's doing a lot of research right now exactly in trying to figure out the sequences and things, so if students are interested, they should.
Yeah, so just a quick shout out to Dr. Bartlett. This is exactly what he does.
Yeah, he's studying.
He's doing all of this, particularly interactions with the polymerases and all of the general transcription factors, but in a way cooler organism than eukaryotes. This is Pyrococcus furiosus and arcan. So, quick shout out there. So that is, again, the overview, holoenzymes, what's happening. Most of the time, what you're doing is trying to get the polymerase to the promoter. And this is an overview here of all of the possibilities that you can have in terms of transcriptional activation. And this is your sort of original activation process. What we've talked about so far has all been here with B. So most of the time, your transcriptional activator, which is this guy right here, interacts with some component of the holoenzyme, gets it here, you get transcription. And again, I would say 90% of the promoters are being regulated this way. Many cases, your single gene activator is not enough. It has to interact with other transcriptional activators. And so very often, we talked about the cooperativity. One of these is going to give you 1 unit. The other one will give you 2 units. Both together give you 100 units in terms of transcription. So this is another very common occurrence. But usually this will be, you get both of these together, and then this happens here. In a few cases, you have transcriptional regulators that will bind to sequences either right next to the polymerase or an enhancer a long way away, which will now activate some part of the polymerase in order to get it to start transcribing. And a classic example there would be stimulating the activity of TF2H. TF2H does what? Helicase and kinase activity, exactly. So the helicase is pulling the 2 strands apart, so you can actually start transcribing, and the kinase is going to phosphorylate the C-terminal domain. So if you stimulate that activity by one of these activator proteins, clearly this could be going on. The last one that we didn't really talk about very much, but it's a more recent kind of thing, and there may be quite a few promoters that do this, is that the RNA polymerase will actually start down here at the bottom and get a little way away from the core promoter, which is basically shown here. You see the RNA polymerase has moved away from right where it starts, and then it just sits there. This is a pause. So this is not attenuation. Attenuation is where that polymerase completely comes off. It terminates. Here, it's a pause, and your gene regulators can break that pause and allow it to continue transcribing. And so this is going to be a very rapid way to get transcriptional regulation, because if you don't have to get all of this stuff to the promoter, which of course you've got to do in terms of getting rid of all of your nucleosomes, etc., if you've already got a polymerase primed and ready to go, this is going to be a much faster way to get your gene regulation going on. Yeah?
While it's sitting there and waiting, is it doing abortive initiation at all? Or is it just...
So abortive initiation, so the question is about abortive initiation. Abortive initiation is usually thought of in this step here. So this is going to be considerably longer than that. Usually it's tens to up to about 100 nucleotides. Abortive initiation is usually 5 or 6 nucleotides. Okay, I talked about these, co-activators, co-repressors. Again, these interact with DNA binding proteins through protein-protein interactions and can activate or repress transcription. That activation can be direct, i.e. it binds to some part of the holoenzyme and or mediator complex, or these can be things that are interacting with chromatin. And much more often, these are things that are interacting with chromatin. Which brings me to another thing that we've had a lot of questions about, open versus closed covered promoters. So an open promoter just means there's no nucleosome bound where the holoenzyme is going to be binding. So the core promoter is available for the polymerase to bind to it. And this is true for basically non-regulated genes. So the particular genes for proteins that you need all the time, ribosomal protein genes, RNA polymerase genes, etc., things that you need for basic metabolism. So these are going to be the open promoters. They'll bind right here. Curiously enough, very few of these actually have Tata boxes that are associated with them. And that also tells you they don't need as many of those general transcription factors in order to get them bound there. Interesting question how you get the polymerase to these places. It's not actually really well known how that happens. They do have lots of A's and T's, which probably helps the two strands to come apart. And you have, again, here down at the bottom, very little regulation. On the other hand, of course, the covered promoters, those that have nucleosomes associated with them, those are going to be your regulated promoters. If you look at the regulated promoters, how do you get to those? It's usually a multiple-step process. So you've got your core promoter, which is just represented on this slide as the Tata box associated with nucleosomes. So somehow you've got to get your holoenzyme to that Tata box. And remember, it's a bunch of proteins. There's a whole bunch of stuff here, so it's way bigger than the nucleosomes. So you have multiple steps here, usually a gene activator protein, some kind of histone modification enzyme, and often multiple modifications of your histones. And then those histone modifications will bind to parts of the TF2D complex. And that binding process, in particular through TF2D, happens through these things called bromodomains. And bromodomains, I again notice this slide, didn't talk about them before. A bromodomain binds to acetylated lysines. And so lysine acetylation is very often leading to dissociation of the nucleosomes from the DNA, but also provides a binding site for these general transcription factors, which will lead to the assembly of the whole process. Yeah?
Is there a bromodomain in this diagram?
Yeah, so the bromodomains are specific, protein structures which will bind to certain things. And so this part of TF2D right here, there's a bromodomain which is binding that acetylated lysine. There's a bromodomain which is associated with this acetylated lysine. There are two kinds of domains that are like this in terms of associations with your modified histones. There's also a chromodomain. And the way I remember chromodomains is that's important for heterochromatin, which is going to be on methylated lysines. So chromodomains, methylated, heterochromatin, bromodomains, the other one. So somehow this projector eats some of my images. I'm not quite sure how that happens. So again, one of the areas where there's been lots of questions. What about this crazy? Oh, sorry. Back.
Are the bromodomains and chromodomains, are those always on histone tails?
So the question is, are the bromodomains and chromodomains always on histone tails? No, they're domains that interact with what things that are on histone tails. So they're not the histone tails themselves. Those are just ones that get modified. But then those will interact with these modified histones. So that's where you have bromo and chromodomains. And again, a quick aside here, if you think about some of the chromatin remodeling complexes, not surprisingly, these are going to have bromo and chromodomains as well, because they're going to interact with these modified histones. Okay, so this image here was the one that had, mamma mia, these slides are just going completely nuts here. So the activation parts of the even skipped. And so I'll go over these relatively quickly here. I could diagram them up on the screen, but that would take us to the end of class. But the idea here is that we've got a particular regulatory sequence, about 20,000 nucleotides in Drosophila, which causes the expression of one gene, and that gene is even skipped, EVE, in 7 individual stripes on your Drosophila embryo. Yeah, Travis.
As far as technically, I've noticed if I go from just the normal non-slideshow view of this,
Yeah.
the images come out.
Yeah. So, want to give that a try? Let's see. No, they're dead. Yeah, so, yes, they've got this big X. They were fine this morning on my computer when I looked at them before class. So, yeah.
Can you kill some processes? It says you don't have enough memory.
I can kill something. I could, no. Actually, let's try killing Safari. Maybe that'll help. Try it again. Okay. No. Yep, it doesn't like me. I'd probably have to restart everything here. But the basic message here, and so we can sort of imagine, just imagine your Drosophila embryo with the beautiful 7 stripes of even skipped on it. But the idea here is that, and it's a slide. Hopefully, maybe even some of you have them in your notes if you printed them out already. So those it's there, right?
Yeah. They're on ours, just not on yours.
Go figure. But I just obviously need a new computer, right? Someone to buy one for me? So 7 stripes, and those 7 stripes are regulated by this 20,000 nucleotide region. If you take just a 500 nucleotide segment of that, it's the stripe 2 regulator, and hook it up to a reporter gene. We didn't talk that much about reporter genes, but I did want to mention it again here. Reporter genes are, again, one of these genetically engineered constructs that you have a gene that produces something you can detect. In this case, it's beta-galactosidase, which will bind to and hydrolyze something that's otherwise colorless and turn it blue, called XGAL. If you express that gene in the embryo, that'll give you, wherever you have expression of that gene and that activity, will give you a blue stripe, or blue anywhere. And the fact that you have the regulatory sequences in front of it, which are deriving the expression of that particular gene in that particular stripe, that tells you with your reporter gene, you just need that particular sequence. So 500 nucleotides of sequence to there. And then if you look at that 500 nucleotides, what you see is that it binds to, obviously, 4 different proteins, which are these guys over here, bicoid, giant, hunchback, and cripple. And the way you do that is by doing one of these band shift experiments. So EMSA. You have that one particular 500 base segment. You put it together with these 4 different proteins. You see that they all bind to this particular piece of DNA. Now, the other thing that you know is where these proteins are being expressed in the Drosophila embryo. And you know that bicoid and hunchback are activators, giant and cripple are repressors. And these are nice and simple. They don't act by interacting with chromatin remodeling complexes. They're just competing for binding to that particular piece of DNA. And you know this through all of your DNA binding experiments, et cetera. And so what you get, lo and behold, we have figures again, is in just this one place in the embryo, you have the presence of 2 activator proteins, but none of your repressor proteins. And so what that is this little area right here in your Drosophila embryo where you don't have repressors, you still have activators, so you're going to be expressing that particular gene in that particular location. So it's an example of combinatorial control because you've got activators and repressors, which are there. Anytime you move out of that particular region, which is what you see here, you've got repressor protein. That repressor protein is going to stop the binding of your activator protein. You don't get expression. You move posterior to that area, you have this next repressor protein, which is also going to be blocking expression there. And so this is really an example of combinatorial control, a very simple example of combinatorial control. The same thing happens at each of the other 7 stripes, only there are going to be different proteins that are involved in each of those. So it's just a way of getting different expression. Yeah?
So the giant retrieval and all that, that's just restricting the Victorian.
Right. So if you think, so we've got the whole regulatory segment, the 20,000 nucleotides, is going to bind to lots of different proteins. We're just looking at what's being regulated by that 500 base piece of that 20,000. And what binds to that 500 base pair piece are just these 4 proteins. So how do these enhancers work? Of course, it works by looping. Looping is, of course, a problem if you've got a big genome and you're interacting here. You've got a gene activator protein. You want to make sure it only activates one particular core promoter or a set of relatively small number of core promoters. But if it's looping, how could it be not interacting with all of the other promoters of the 70-odd thousand that you happen to have in your genome? So the way this happens is through the activity of insulators plus insulator binding proteins. And so that's the real critical aspect here, is that just having an insulator, which is a DNA sequence, does nothing for you. You have to have insulator plus your insulator binding protein in order to isolate the particular region which is between these insulator sequences. If you have that, you have this insulator binding protein, then this regulatory region right here is only going to interact with other genes that are in that particular part of the genome. This is why when you have imprinting, in terms of the IGF-2 gene, that allows activation beyond the insulator sequence because the insulator is methylated. If the insulator is methylated, the insulator binding protein doesn't bind to it anymore. And that's usually what's happening with DNA methylation. So DNA methylation, eukaryotes, bacteria is a different story, but in eukaryotes it's almost always at the 5 position of cytosine. You have methylation right here. That methyl group is sticking out in the major groove. Where do your DNA binding proteins associate? In the major groove of DNA. So methylation is going to change binding. And there are, it turns out, some proteins that bind specifically to methylated DNA, like the maintenance methyltransferase, that will bind to methylated DNA on one strand and say, hey, we need to methylate it on the other strand as well. So this, we've talked about DNA methylation for binding of specific proteins, again, like these insulator binding proteins. The other thing that DNA methylation is usually used for is gene silencing. But gene silencing you don't just get with DNA methylation. It's a whole combination of different modifications to the DNA through methylation, but also modifications to the histones and binding proteins that are going to bind to the modified DNA as well. So you really need 3 things to have this work properly. You first have your methylation enzyme, which starts here, methylates some of your histones. Then you have one of these read-writer complexes. What kind of domain is a read-writer complex going to have that binds to methylated histones? A chromodomain, exactly. So chromodomain containing proteins that are also histone methyltransferases will bind. They'll methylate all of your histones. Methylated histones also interact with DNA methylases. These DNA methylases, of course, are going to methylate at the 5 position of your cytosines. And then those have methylated DNA binding proteins. So it's not just the methylation of DNA, but also the binding protein that binds to those methylated DNAs, which really causes the complete compaction of the DNA in something like the inactivated X chromosome. Yeah?
So is the chromodomain like the writer?
So the chromodomain here would be the reader, not the writer. The writer would be the histone methyltransferase. So if you have a protein that's got chromodomains and histone methyltransferases, that could really easily be a read-writer complex. Yeah?
And we're calling the whole protein a chromodomain or a bromodomain, not a particular domain of the protein?
Okay, so this is a bit of terminology issues here. So the whole protein is not going to be one domain, so that chromodomain is going to be a domain. It's a part of the protein. And so that particular part of the protein, and then the histone methyltransferase. So if that would be the reader part, would be the part that has the chromodomain, and the writer part is the one that has histone methyltransferase on it. Yeah? Yes? Could I say what they are? And they'll be on tape, so you'll know them and can study them for Tuesday. First one is going to be histone methylation. Second one is going to be DNA methylation. Third one is going to be your methylated DNA binding proteins. Got it? What are they? Oh, what are the question? What are the three things that you need to get full silencing? You need histone methylation, you need DNA methylation, and you need a methyl DNA binding protein. They're the three things that you need to get full silencing. Yeah?
What's the difference between when do you call an enzyme a methyltransferase and when do you call them methylase?
So I try to call, so the question is, when do you call it a methylase and when do you call it a methyltransferase? Usually, and I probably mentioned this before, enzymes, ACEs, usually what they do, they cut stuff off. And so usually when you think of an ACE, it's going to be breaking something down. Now here, of course, you're adding a methyl group. And so that's why I like to call them histone methyltransferases. They do kind of get used interchangeably, and as you can see here on the slide, actually just uses methylase. But in order to try and be more specific, I try and use the methyltransferase. Okay. Quickly, genome imprinting. Message here, there was some confusion about maternal versus paternal imprinting. As far as we're concerned, i.e. what's happening on Tuesday, imprinting is just about methylation. And so here, imprinting is happening on, and this is just a case, you also have methylation that happens in female lineages as well. Here, you have imprinted alleles in both mice. These are the ones that came from the father in this particular case. When you undergo meiosis, methyl groups are all removed, and they're actually removed everywhere. And then you have this specific methyltransferase, getting back to your question in the back here, which will methylate this particular allele, gene, insulator element just in sperm and not in eggs. So now the offspring are going to have the two different alleles that came here from the male because, diploid organism. So here we've got one of those alleles which has been methylated. Here we have the other allele which has been methylated. Yeah?
Is that typically carried through like the whole species? Like this species will typically have males, methylated at this gene?
So I guess the question is this species specific?
Or does it vary between individuals?
Does it vary between individuals? Yeah, it is pretty species specific. And so the question is really, again, now, in mice, you will see imprinting and methylation at this particular position. In humans, you'll see methylation imprinting at different places. And so it's vary. And it's interestingly enough, you find it pretty much only in mammals. It seems to be a relatively recent evolutionary process, how this happened. Again, quite why that is not entirely clear. If anybody listens to the TWIV podcast, you should never ask the why question in biology. Because there isn't a good answer to why. And we'll finish up today just by going through epigenetics. And epigenetics, again, is just inheritance in the absence of a change in the genetic material. So no change in your genetic sequence. Here, we've talked about chromatin modification. Again, if you have a modified histone, you replicate that modified histone, or the DNA associated with that modified histone, because of the read-writer complexes that will also modify the other histones and keep them in the same way, even though that genetic material hasn't changed at all. Same thing is true of modified DNA. Now, the modification that happens on your cytosines is going to be, that one particular modification because of the methyltransferase which is there. It'll still give you that methylation. And then we also talked about the positive feedback loops here. Once you have expression of a protein which induces the expression of that same protein, that will lead to that particular state being inherited through time. Good luck on the final. Thanks for a fun term.